Please welcome ASM Chief Executive Officer Stefano Bertuzzi.
Good evening, everyone, and welcome. Welcome to the President Forum. I hope you're having a great time at the Microbe meeting. If you're having at least half the fun that I am having, we're in a really good place. I have been so energized by meeting so many people, so many young scientists, and the conversation has been so fantastic. Look around, it's been a wonderful, diverse crowd with us, and this is so, so beautiful to see, very energizing. Microbiology is in a good space because of you, and after this meeting, I think we can do even more. So let's give ourselves a big round of applause, everyone. Leading us tonight at the President Forum will be our President, ASM President, Dr. Steve Finkel. And I would like, on behalf of the whole board of directors, really thank Dr. Steve Finkel for his resonant leadership. With Steve at the helm, the organization has grown, the organization has taken on new challenges, has expanded its reach in the country and outside of the country. And we're really so happy and so fortunate to have you, Steve, as our president. So join me and welcome on stage Dr. Steve Finkel.
Thank you, Stefano, for that very kind introduction. I really do appreciate it. So good afternoon, everyone. I'm delighted to see you all here today, and I can actually see you. The house lights are up high enough for me to see you. One of the traditions at ASM Microbe is that the president hosts a session on a topic of their choice at the meeting. And today I'm delighted to participate with you in a session focusing on the changing human microbiome. Now, of course, the quote-unquote human microbiome has, without question, experienced significant changes over the course of human history. Undoubtedly, the advent of agriculture, the development of human communities that are ever growing in size, and industrialization have had major impacts on human-associated microbial communities. Depending on your particular area of research, the kinds of changes, the kinds of phenotypes associated with that change can be very different. When I think about my own studies of the gut microbiome, with a focus on fecal microbes that generate electricity, and yes, it is happening inside all of you right now. Your microbes are making electricity in your gut. Paper coming soon. Changes in that microbiota have very interesting consequences. And just thinking about the title of Joe Handelsman's new book, A World Without Soil, one cannot but immediately wonder how that could impact the human microbiota, as well as many other aspects of life. Or maybe you are simply interested in living the healthiest life you can and want to understand more about those changes. No matter the context, I think that we can be fairly confident that the rate of change humans are currently experiencing is likely faster than ever experienced before. And each of today's three panelists is studying an aspect of this change, and I very much look forward to hearing from each of them. Our first speaker will be Dr. Justin Sonnenburg. I'm a professor of microbiology and immunology at the Stanford University School of Medicine. His research is uncovering basic principles governing interactions within the intestinal microbiota and the host, with a particular interest on the effects of perturbations of that gut microenvironment. These perturbations can range from changes to the input microbiota themselves to changes in the physical-chemical environment, including changes in diet and environmental stressors. Our second speaker will be Dr. Gloria Dominguez-Bello, the Henry Rutgers Professor of Microbiome and Health in the Departments of Biochemistry and Microbiology and Anthropology at Rutgers University. She also serves as the director of the New Jersey Institute for Food, Nutrition, and Health. Her research focuses on the development of the human microbiome from birth and the impact of changes in the environment, including urbanization, on the early life microbiota transmission and colonization. Our final speaker will be Dr. Eric Alm, a professor of biological engineering at MIT. His research combines computational and theoretical approaches with experimentation to understand the function of the human microbiome, as well as potentially engineering novel microbiota. His work has shown that the genomes of organisms and gut communities are not static, but are constantly adapting through a variety of mechanisms, including mutation and horizontal gene transfer. Now, before I ask our first speaker to join me on the stage, I'd like to explain the structure of today's session. First, each panelist will give a brief overview of their research, including topics of particular interest with respect to today's theme. After all three of our panelists have given their presentations, we will all gather here on stage, the four of us, for a question and answer discussion section. During the Q&A session, I will ask you, the members of our audience, both here in person as well as our colleagues who are participating remotely, to ask questions of our panelists. I very much encourage students and postdocs in the audience to ask your questions. And what we'll do is, when we all come and sit up here, I'm going to ask folks to line up at the two microphones that are here to my right and in the center. Okay, so that's how we're going to run it. And we really want a lot of great questions from the audience. So once again, let me thank you all for attending this afternoon. Also, I would be remiss if I did not also thank my colleague and former ASM President, Dr. Jeff Miller, for helping to inspire today's session. It's wonderful having colleagues like Jeff. We bounced ideas around, and that was really the genesis of this session. So Jeff, wherever you are in the room, I know you're here somewhere, thank you very much. I'm very much looking forward to three very interesting presentations and a very robust discussion. And with that, I would like to now invite to the stage Dr. Justin Sonnenburg to give the first presentation.
Okay, thank you, Steve, for that wonderful introduction, for creating this session. It's really an honor to be here, honored to be with Eric and Gloria in the panel, and great to be with all of you. Thank you all for coming. So of course, my lab studies the gut microbiota or microbiome, shown here in this cross-section of a mouse colon. You can see the microbes in the top left, all these colorful little rods packed up against this green mucus layer overlying the blue epithelium. So you can immediately see how dense this community is, how close it is to our tissue, incredible interface for exchange of biological information. And so we harbor, of course, trillions of microbial cells. Incredible coding capacity is embedded in this community. And my lab is really interested in understanding both who is there, what microbes live in our gut, and we're actually still in the process of mapping this because we focus so much of our attention as a field on industrialized microbiome. So there's still a lot to do just in taking the census of the gut microbiome. And then we're also interested in what these microbes are doing, how they integrate with our biology. And you know, we've known for decades that microbes in our gut are important for gut health. They're important for digestion, but an incredible body of work over the past 10 to 15 years by a huge number of investigators has established that our gut microbes are actually doing much more than this. They're impacting our metabolism, our immune system, potentially impacting things like neurodegeneration. And you know, I think that while this, you know, image depicts the microbes as being upstream of everything, perhaps a better way to think about it is that our microbes are really just part of this integrated system that is our body. And if you perturb the microbes, those changes ripple through our biology. And so what's special about the gut microbiome is that beyond it being highly individualized is that it's malleable. We can change it. And when you combine the fact that it's connected to so much of our biology and that it's individualized, this is going to be an incredible mode, I think, of precision health. When we understand the interactions, have the tools and the knowledge base, we'll be able to manipulate these microbes to positively impact our health. But this malleability also represents a vulnerability. And undoubtedly, there's plasticity that's built into the system that's the system's tolerant of plasticity, but there's probably a limit to how much plasticity can be tolerated. And so we think a lot about the asymmetric plasticity of the human microbiome and the human genome over evolutionary time. We have these two components of our biology that have to work together, and yet our microbiome can change incredibly rapidly. Antibiotics, Western diet, C-sections, baby formula. And so these changes impact our gut microbiome, yet our human genome doesn't change over those same timescales. And so could it be that there's somewhat of an incompatibility right now between our gut microbiome and our human genome, that we have somewhat of a foreign entity living inside of us. And so we think a lot about these questions. What were the common features of the microbiome as humans spread around the planet? Were there common features that perhaps our human genome grew to expect to be there, that we co-evolved with? And has the microbiome changed recently? And so to answer these questions, we actually study indigenous populations, groups like the Hadza hunter-gatherers of Tanzania. And I just want to spend a second to note that it's really important to note the importance of ethical research with indigenous populations. Not just making sure that you have the right material transfers agreement with the right language in it, the right permits, but that you're consenting properly, you're explaining the research, and that you're presenting your work back to the groups that you're working with in this research that you're sampling from. And then we're even working with the Gates Foundation, not specifically with the Hadza hunter-gatherers, but with other populations in low and middle income countries to try to facilitate in-country science so that where the samples are collected, that group of people can be involved in the discoveries and both generating and analyzing the data. And so I wanted to start with our first study that we published on the Hadza hunter-gatherers, and that really focused on the seasonal cycling of the microbiome. But the final figure of that paper was a really important figure, I think, the work of this former graduate student, Sam Smits, phenomenal graduate student. And he took our data from Tanzania and brought it together with other microbiome profiles from other populations that had been surveyed to that point in time, and put that all in the same principal coordinate plot where you can look at the relative relatedness of these different microbiomes. And so that data is summarized here, the different populations' gut microbiome composition represented by that bar on this principal coordinate axis. And you can immediately see this really stark pattern: non-industrialized populations on one side, industrialized populations on the other side. And this isn't just a loss of diversity in the industrialized microbiome, it actually is a stereotypical changes in the groups of microbes that live there. So if you look at the fractional representation of different families of bacteria that explain this variability, groups like the spirochetes, Succinivibrio, Prevotella, all very abundant in traditional populations, and they are rare or absent in industrialized populations. Verruca microbia, on the other hand, Akkermansia mucinifila, blooms in industrialized populations. We know that's a mucus utilizer. We know that Western populations eat low fiber diets. So one of the disadvantages of that analysis is that those populations have different host genetics, they live in different geographies. So Ashish Jha, former postdoc in the lab, now faculty member at NYU Abu Dhabi, went to his native Nepal and surveyed 4 populations that live on a lifestyle gradient. So this is just an analysis of an extensive survey questionnaire showing this lifestyle gradient. The Chipang are foragers, the Raji and Raute are populations that settled in the 1980s. They forage a little bit, but mostly farm. And then the Taru are farmers. They've been settled for 250 years. And so Ashish wondered what was happening to their gut microbiome along this lifestyle gradient. And so we surveyed their gut microbiome. And on the left, you can see a summary. We see a direct correspondence in their lifestyle and their gut microbiome relative to one another. So the foragers are on the left, transitional communities are in between, and then we've thrown in some Americans in orange there on the top right. If you want to look at this data in more detail, there's a heat map there. Here, the different rows represent different taxa that explain the differences across these populations. The columns are the different people, and up at the top, you can see the little labels of the populations. And so, you know, if you just look at the foragers on the left versus the Americans on the right, you see this stark differences, very similar to what we saw in an independent analysis of the Hadza. Same bacterial families are disappearing. And then what's nice is you capture these intermediate states of microbiome transition as groups change their lifestyle more towards industrialization. And so we call these the vanished taxa, volatile and associated negatively with industrialized societies of humans. And Ashish actually proposed in this paper a model of different patterns of species loss and gain over this lifestyle gradient. So the vanished taxa, and then also the blossom taxa that either bloom or are selected in societies of urbanization or modernization. And so that's all 16S data. We've also been accruing metagenomics data. This is from a phenomenal group of trainees, Brian Merrill, Matt Carter, Dylan DeHaan, and Matt Olm, brought together, they actually did tremendously deep sequencing in the Hadza and then brought together 1,800 metagenomes from different populations around the world. And they began to plot the prevalence of different species from the genomes they were assembling in these metagenomic data sets across this lifestyle gradient. So this is hunter-gatherers on the left, transitional in the middle, industrial on the right. And we can see these patterns of blossom taxa in blue and vanished taxa in pink. So these are individual species changing prevalence across this lifestyle gradient. And we can see this actually a little bit more cleanly. If we just separate them out, we see dozens of vanished taxa, dozens of blossom species. And what's nice is we actually define these by looking at the extremes, but if you plot the transitional communities in between, in the middle there, you actually see this intermediate state of prevalence. So adding to the evidence that lifestyle is really the driving force behind these changes. This is currently under review and now in on bioRxiv. So we've also looked at the infant gut microbiome. This is actually work that we just published in Science this past week. And we see a very similar pattern that, you know, they're blossom species, they're vanished species, they're different than the adult, but very similar patterns. And the really striking one here is Bifidobacterium infantis, the canonical degrader of human milk oligosaccharides. It's the green line there, is actually one of these vanishing species, so disappearing in industrialized nations. So that's all taxonomic, you know, phylogenetic compositional changes. What happens to function? Are these changes maybe functionally redundant? Well, no, they're definitely not. So if we go in and look at the functions that are enriched either in the blossom or the vanished taxa, we see big differences. So we know that these changes are not functionally redundant. And if we go in and look at the specifics, there's actually a really interesting enrichment in genes involved in oxidative stress in these blossom taxa, in the taxa that are more abundant in the industrialized guts. And so this might be the signature you would expect in guts that are slightly inflamed, where there's simmering inflammation or a different metabolic state. So interesting things to follow up on there. And so how similar are non-industrialized gut microbiomes to ancient microbiomes? This is a really important paper from Alex Kostek's lab at Harvard. They sequenced paleofeces, fossilized stool or coprolites from humans that were 1,000 to 2,000 years old. And then they determined the composition and overlaid that onto present day industrialized and non-industrialized microbiome composition. You can see this very clear pattern that the paleofeces have similar composition to the non-industrialized microbiomes. And this adds to the evidence that the industrialized microbiomes is this recently derived state that deviates from what we likely evolved with. So what microbes migrated with humans around the planet over the past 100,000 years? This is a really important question. We're just starting to look at this, but this is one example. Treponema succinifaciens is one of these bacteria that has gone extinct in industrialized societies, but we see in all of these non-industrialized populations. And so if we assemble genomes of different strains from different populations and then paint that phylogenetic relationship of those strains onto the geography of where these populations are from, we start to see a pattern that mirrors human migration. So it really suggests that as humans migrated around the planet, they were passing these microbes from generation to generation. And then in the evolutionary blink of an eye, as we industrialized, these microbes disappeared. So our microbial identity has changed. What does this mean for us? We know that chronic inflammatory diseases are on the rise. We also know that in animal models, if you change the gut microbiome of an animal, of a mouse model, for instance, you completely change how that mouse immune system functions. So could it be that this foreign microbiome inside of us is somehow dysregulating our immune system and driving us towards inflammatory and metabolic disorders? So can we repair our microbiota and improve our health, or at least test if health can be improved via microbiota change. I want to provide some ray of hope here. So we started the Center for Human Microbiome Studies at Stanford, and the goal of this was really to turn the biomedical paradigm that I had learned for how to do science on its head. So instead of working from molecular mechanism through animal models and trying to translate to humans, starting with experiments in humans, seeing what the findings are, and then reverse translating those into mice. And this is largely possible because of incredible partners that I have on this slide, and also because we're doing dietary interventions. And we know that diet is assumed to be safe in humans, and we can just start doing trials in humans right away. So I just wanted to provide a flavor. We've done many studies like this, but this is one of the more interesting ones in my mind. This is what we call the FIFO study for fermented fiber food study. And the question really was, can we modulate the immune system via microbiota targeted diet? So we took people, enrolled them, surveyed them at baseline, ramped them onto a high fermented food diet. So eating kombucha, kefir, yogurt, kimchi, 6 weeks of this high fermented food diet, and then a washout period, all the while collecting stool and blood samples for comprehensive microbiota and immune profiling. I want to point out that Maddie Topf, Hannah Wastik, and Gabby Fragidakis are all former trainees that really drove this study forward, and it wouldn't have been possible without phenomenal collaborators in Christopher Gardner's group, really phenomenal group to work with. Dahlia Perlman is really the key. She's the one that got people to eat differently. We call her the diet whisperer. And so this is what she accomplished. So she got people up to, during the maintenance phase, over 6 servings of fermented foods per day. And so the question is, what happens to their microbiome? Well, the microbiome goes up in diversity. And so what happens to the immune system? Well, over this same time, many inflammatory markers decrease. And I'm just showing 4 here. There were actually over 24 different inflammatory markers surveyed through 2 different means, CyTOF and O-links cytokine profiling. We saw many markers of inflammation decrease. So this exactly the signature we would hope to see: increasing diversity, decreasing inflammation. The problem is that we're not bringing back vanished species, and we don't really have a deep readout of health. We just have these immune markers. So we need to do these for much longer periods of time. Now, we also did this with a high fiber diet, and we didn't see the same changes. There was not the increase in microbiota diversity. The immune story is a little bit more complicated. And it really is consistent when looking at the literature. So if, this is an important study out of Dan Knight's group, where they tracked immigrants from Southeast Asia to the United States and saw within months their microbiota started to industrialize, lose Prevotella species, lose diversity, increase Bacteroides. The longer they were here, the more profound the changes became. And what's important here is that they lost fiber degrading microbes and enzymes. And so the Western microbiota is lacking the capacity to degrade dietary fiber largely. And so it's no wonder when we feed people over the course of 6 weeks a high fiber diet that there's not big changes. We saw big changes in the small group that had the highest diversity, and that's an important nuance. And then just to finish up, I wanted to go back to a multi-generational mouse experiment that we performed several years ago. This was just looking at the impact of Western diet versus a high fiber diet on a mouse microbiome, asking the question of whether a low fiber diet could drive extinctions in the gut microbiome. And that's exactly what we see. So we see that over the course of 4 generations, compounding extinctions, elimination of dietary fiber depletes the microbiome. And when we put those 4th generation mice back onto a high fiber diet, this is asking the question, is it too late for us? We actually don't see recovery of that diversity. So all those microbes that are extinct stay extinct. When we do a fecal transplant from the 4th generation high fiber fed mice, all the diversity comes back. So microbes plus diet can reconstitute some of the diversity that we've lost. So this is where we're at. We've changed our microbiome through lifestyle. Where do we go from here? How important is this for our health? Is dietary change enough to bring back health in a healthy microbiome? Do we need to reintroduce species? These are all burning questions in my mind that we hope to answer over the coming years. I want to finish by thanking my incredible group. many wonderful collaborators. I want to specifically point out Erica Sonnenburg, my wife, who helps run the research program with me, and Stephen Higginbottom, who runs our Notobiotic group, really phenomenal group of researchers. Thanks to all of the groups, all of the foundations that have funded us, and thanks to all of you for your attention.
Please welcome Dr. Gloria Dominguez-Bello.
Good afternoon, everybody. It's a great pleasure to be here with you, and it's an honor. Thank you, Steve, for inviting me. And I think it has been a great start for my talk because I can go fast in the introduction. I'm going to touch two topics. One is restoration, and the other is preservation for future restoration. So as Justin mentioned, there is a rocketing increase in immune and metabolic diseases. And there are two hypotheses that try to explain this phenomenon, and both are dealing with microbes. One is the hygiene hypothesis related to environmental microbes and lack of exposure. And the other is the disappearing human microbiota hypothesis related to lack of human microbe diversity acquisition. But this really reflects the environmental exposures naturally in life. From birth, the human, highly human maternal environment that babies have until the brain develops enough, the immune system, sensorial, motor, and then babies start exploring heavily the environment. And in our society, we perturb each step of these exposures with antibiotic C-section, antimicrobials, technologies that separate mothers and babies, raising kids in clean apartments in urban cities far from nature. I study C-sections, and we know that C-sections lead to different trajectories of the fecal microbiota in kids, and we can see that here in a 24-month period. And this happens in humans, but also in mice. So we are studying mice phenotypes to understand what happens and what are the changes. And we have, this shows metabolic effects induced by C-section. And you can see that not in males, but in females, we have the three groups, the vaginally born, the C-section born, and then a group of C-section plus restoration with vaginal fluids of the mother. And what you can see is that the females have lower nocturnal metabolic rate if they were born by C-section, and restoration doesn't do much, maybe an intermediate phenotype. When we look at glucose intolerance test, we can see that the pink vaginal birth is lower than the blue C-section, and the C-section babies that were restored normalized. So there is higher glucose intolerance in C-section born that were not seeded or restored. We also found differences in mouse activity. So the pink vaginally born males and females have higher nocturnal activity than the C-section born, and vaginal seeding maybe we are under power, but maybe partially restored in females. So this is remarkable, the change in behavior. And this is a paper by John Pryan, where he studies the behavioral effects of C-section. So we decided to go to the brain and study brain proteins. And this is in collaboration. Jeremy Lessing is a student that I share the tutoring with Manny Di Chico. Jeremy is studying proteins in the proliferation, important in proliferation of the brain, cytoskeleton or differentiation. And he found differences in the cerebellum at day 7 after birth. There is higher proteins like cyclin E, and in the cytoskeleton TUG1. He also is finding changes in hippocampal proteins. This work is ongoing. Then we ask, if we have babies born by C-section or vaginally, what is really the functional relevance of their microbiota? And we can test that putting that poop into mice, into germ-free mice. And we are doing this study in collaboration with Liping Zhao at Rutgers, and we are only starting the data analysis. I'm going to show you the inoculum and the microbiota of the mice at 4 weeks. This is showing the fecal inoculum for 3-month-old babies. And you can see how discordant they are in the principal coordinate analysis. On the bars, on the taxa plots, you can see the different colors, in particular, the low bacteroides or no bacteroides in the C-section is highlighted. When we look at the offspring, what we observe is that if the mice received poop from babies born vaginally, they engraft better in relation to the C-section poop. They have higher alpha diversity, and you can see on the right, the principal coordinate analysis really clearly showing different trajectories of the poop in the mice from babies born by C-section or vaginally. We observe there is also higher transfer and higher engraftment of the feces from babies born vaginally in the mice. And the distance between this offspring and their mother is closer, so there is higher fidelity also. From the study of T-cell profiles in collaboration with Derek Sanangelo, I'm going to show the significant ones. He's studying 3 tissues, lamina propria, lymph nodes, inguinal, and the spleen, and we can see differences. In particular, activated CD4 plus is higher in the vaginal, in the mice receiving poop from vaginally born kids. Th1 is higher in the C-section group. Th17, interestingly, which is pro-inflammatory, is higher in the 3 tissues in the vaginally born. There are more differences of other proteins. In the inguinal lymph nodes, you can see higher CD4 plus and higher FOXP3 in the vaginal poop, in the mice transfer vaginal poop. So in summary, in relation to mice transplanted with feces from vaginally born babies, those that receive poop from C-section born have different microbiota, but also different immune responses. So these poops are different. Can we restore? And we did the first observational clinical trial restoring C-section born humans with the vaginal fluids of their mothers. We published a bigger observational study with 174 families, and using compositional tensor factorization analysis, which reduces dimensionality and produces much nicer results, we could see that in relation to the blue trajectory in feces, in mouse, and in forearm, the red C-section babies have a different abnormal trajectory. And the green, when the babies born by C-section are restored, exposed to vaginal fluids of their mothers at birth, you can partially normalize feces, not entirely, but you can normalize pretty well mouth and skin. In this study, we realized, we found that there are perinatal changes in the vaginal ecosystem that have not been very well characterized. On the left side, you have the microbiota of a non-pregnant women from the Human Microbiome project. And you can see that the sources is mostly only vaginal, not shared. There is a 20% of sharing with other body sites. But on the right-hand side, these are women giving birth. At the time of giving birth, more than half of the bacteria you find in the vaginal canal of women are shared, mostly with more than one body site. And you can see clearly fecal bacteria, oral bacteria, and nasal bacteria there. So the vaginal microbiota perinatally is a pluripotential organ ready to colonize, to provide colonizers to all body sites of the babies. I did my homework, and Steve asked us to say our optimistic view of the field in 2040. Doctors and the public are going to be more knowledgeable about the microbiome, and therefore there will be less antibiotics and less C-section use. After new and very important research that we have not done yet, women will have improved experiences giving birth vaginally. We haven't done research on how to improve natural birth. I predict also that some advances will happen, transplanting microbes instead of specimens, but I'm not that optimistic, especially considering where we are with blood transfusions still. And I foresee that people will be doing a lot of autologous transplants. When you're healthy, you keep your microbiome in the freezer. When you need it, you use it after antibiotics, etc. So breast milk is highly restorative. It's anti-obesogenic, and this is a famous paper by Gilman showing how it protects against overweight in adolescents. But in our modern society, because of the unresolved issue between career and maternity, we are still stuck there. The new gold standard of raising babies is expressing breast milk. However, we have circadian rhythms in our blood, and mothers, like everybody else, peak melatonin in the serum at night, as you can see in the top, and peak cortisol, for example, in the morning. And my student, Melissa Wordman, is studying these cycles in hormones in the breast milk. And she's finding that cortisol peaks in the morning in the breast milk and melatonin peaks at night. So therefore, we could be impacting babies by expressing the milk and feeding it out of time. So in relation to the normal feeding where the baby is drinking circadian cues in addition to the very complicated milk, expressing and feeding asynchronously may perturb the microbiome and development. My views, we will still be unable to produce a good artificial breast milk in 2040. There will be better policy for baby health, equity, reproductive rights. Breastfeeding will be acceptable in public in the United States. Every office building will have a baby-friendly space, a daycare at work where mothers can breastfeed and bond with their babies. Finally, the microbiota diversity for future restoration. This slide shows the diversity of cultures, social practices, food, diet, and medicine converging into one single Anthropocene lifestyle. And this changes people's metabolism. This graph shows the urban gradient differences in the urine metabolites of Piaroa Amerindian women. This is one ethnic group distributing from the jungle to a little town. And you can see on the left, the plot by traditional diet, moving from left to right, from high traditional diet to low, and on the right by exposure to urban life. So it's not surprising that given the compounded effect of successive stressors during development, antibiotic C-section formula, hygiene, antimicrobials, urban environments, we see the association between urbanization and gut microbiota diversity depletion, as it was mentioned before. Then we ask, how narrow does the gradient need to be to see an effect? So we did this experiment, this observational study in the jungle with indigenous people that live in the jungle, no market economy, but the difference was that the low urban exposure were extremely remote and rarely visited by doctors. And the medium, we're still living in the jungle, same diet, lifestyle, but we're visited by doctors. That means we're exposed to medicine. And we use the US as a reference population. The study was done in the south of Venezuela. You can see here the pictures of the low urban villages or the medium, and you can see some transculturation in the architecture in the medium. And we did observe the fecal microbiota diversity was lower in the medium exposure in relation to the low exposure. You can see that in the PCOA. The differences were not enough to be close to the Americans, as you can see in the lower graph. Amerindians versus US still keep segregated. We can tell what bacteria are disappearing or not, and Justin mentioned mostly firmicutes and methanogens go down, and Bacteroides and Proteobacteria go up. We can tell the fecal gene content changing, and basically we reduce methane greatly, increase antibiotic resistance, increase sphingolipid metabolism, and increase vitamin metabolism. This is the comparison between the two levels of urban exposure in the jungle, but when we take all the Amerindians and compare to Americans, exactly the same metabolic pathways are, so this is very consistent. In summary, in a population with shared ethnicity and lifestyle, we observe that there is already a reduced microbiota diversity, altered taxa, and changing in metabolic pathways. So because industrialization is degrading the microbiota, and this has been associated with rocketing diseases, can we restore? And for this, we need to bank microbes. We are moving ahead the initiative of the Microbiota Vault. You can look at online. And the core model is local collections that will request a backup deposit in a central collection. Every specimen will be sequenced and the data shared with the world. My optimistic view is that we will value more preserving human traditional practices. We will be better cultivating and analyzing data, but probably not yet at the level of personalized microbiota restoration. And finally, microbiome studies will be more representative of the whole world population. I thank you for your attention.
Please welcome Dr. Eric Elm.
Well, thank you, Steve, for the invitation, and thanks to the organizing committee. This is really an honor to be here. It's especially special to me because the topic I'm going to talk about today, horizontal gene transfer, is something I got really interested in after my very first attendance at the ASM General Meeting. I saw Howard Achman in New Orleans give a talk on horizontal gene transfer and how he was looking at that by running blast on different microbial genomes. And I said, that is absolutely fascinating. And throughout my career, every chance I've gotten to look at bacterial genomes, the first thing I've asked is, who are they exchanging genes with? And so kind of have a confession to make to Steve. When I saw the talk, which was the Changing Human Microbiome, in my head, I was thinking about those genomes changing, those individual microbes in the microbiome changing, because it's been the topic of a lot of my research. And I kept thinking, but he invited me and Justin and Maria Gloria. Why didn't he see the common thread between us? And now I kind of get it. It's the changing microbiome across our whole population. So my talk is going to be more about what's changing inside an individual microbiome, and specifically what's changing inside these genomes. So if we go back, I'll tell you about really the first study that got me really excited about horizontal gene transfer that came from my own lab. And so we had read this paper, it came out in Nature, and they were looking at some genes in algae that help them produce these algal polysaccharides. And what they found is that those genes had been stolen from the algae by some bacterial species that lived in the ocean. And so it was horizontally transferred into those bacteria, and it allowed those bacteria to eat the algae. So that was pretty clever of them. But what they found is that it was also present in a few bacteria that were isolated from the human body. And they found by looking at the phylogeny, looking at the sequences, they were able to figure out that the genes had come from the algae into the marine bacteria and then into the bacteria that were in the human gut. And every single one of those individuals that had a bacteria that had these genes was Japanese. And they have a lot of seaweed in their diet. And I said, that's absolutely fascinating. What if we change our diet, not just the species that are in our guts could change, but the species that are there could pick up new genes and therefore new abilities. And I thought this was the most exciting thing that I had ever heard of. I had two rotation students in my lab, Mark Smith and Chris Smiley, and I said, take all the genomes in GenBank and blast them against each other. And if you see any stretches of DNA that are 1,000 base pairs and 100% identical, that means that that DNA has transferred from one species to another. And I was hoping that we could find another case like this. And they came back a week later and they said, Eric, we did what you said, and we found 80,000 examples for you. And I said, that's crazy. So I enlisted Jonathan Friedman, who was a more senior grad student, to figure out what they had done wrong. They hadn't done anything wrong. And so we were able to come up with this map of what different bacteria are exchanging genes with each other. So here it's showing for every pair of bacterial genomes, what's the probability that they share one of these 100% identical stretches of long DNA. And what you can see is pretty much everybody is sharing genes with everybody, but there's a hot spot around bacteria isolated from the human body. And in particular, if those two bacteria come from the same body part, they're both from the gut or they're both from the mouth or the skin, then they tend to be sharing even more genes in common. So that was kind of our first foray into it. Yara Oren was this graduate student who read my paper. Yara read all my papers, and she would interrupt students in the middle of our group meeting and say, hey, you didn't read Eric's paper. Yara was amazing. She wasn't my student. She was Tal Pupko's over at Tel Aviv University. But she called me up one day, and she said, I read your recent paper, and you really missed the whole story. And I said, oh, really? We were really happy with ourselves. And she said, no. You were looking at stretches of DNA that were at least 1,000 base pairs, and all of the action is happening under 500 base pairs. And she flew out from Tel Aviv University to my lab for a summer and definitely proved that I had missed the big story. And there was even more transfer of the short regions in front of genes. In fact, the housekeeping genes, the most conserved ubiquitous genes, rarely transferred at all, but those same genes had tons of transfer of their upstream regions. So like the non-coding DNA that had all the regulatory signals in it. And that turned out to be one of the more exciting papers that had come out of my group. Ilana Brito is another trainee who worked with me and kind of continued this story. So I actually met Ilana jogging on the Charles River. She was quite fast. I certainly knew she was determined when she told me she was a scientist. And she was talking about going out to Fiji, and she was doing some work there. And I said, wouldn't it be interesting if you could add a microbiome component to your work in Fiji. And we had talked about this for a while. Eventually, she decided to turn most of her attention to the microbiome. She wrote this elaborate fellowship proposal to go into a few villages on the island of Fiji, collect not only microbiome samples from everybody, but see what livestock they touched, collect samples from livestock, collect samples from the drinking water, map who used what drinking water, collect samples from the soil outside their house, collect the GPS coordinates of all the houses, collect the family trees, and then also map out their social network. So she really established kind of all the routes of transmission in the entire community. She wrote this proposal, sent it to NIH, and of course they said, this is much too ambitious. There's no way you can do this. Why don't you scope it down and resubmit it next year? Instead, she put the entire study on her personal credit card and did it right away. She brought the samples back. We had no way to sequence it, but at the end of the year, the Broad Institute had come under budget on one of their grants, and they had a lot of sequencing capacity left. So we were able to publish what I think was one of the largest sequencing data sets by just going through her samples. And what Alana did is she got me into single cell genome sequencing. And I thought this was going to be the answer, and this was going to solve all our problems with horizontal gene transfer, because we could sequence hundreds or maybe even thousands of genomes at once, and we could find where the genes had been transferred. It turned out this was pretty difficult. We spent a long time combining the single cell genomes with a lot of metagenomics. And we were able to reconstruct some of the horizontal gene transfer, but maybe not as much as we had hoped. But the one interesting finding we found is that there were many genes that were unique to, let's say, the industrialized populations or these more rural populations, okay? But there were also genes that were unique to each village. And then there were also genes that were unique to individual people. And so I really started thinking, is it possible? In our first study, we knew there's horizontal gene transfer happening across bacteria that are isolated from people, but none of those bacteria were isolated from the same person. And this is when I really started thinking, well, what about bacteria? What about horizontal gene transfer within a person? So let's say, for example, I eat a lot of seaweed, or I don't eat a lot of seaweed. Then I start eating seaweed, and the microbes in my gut might pick up genes that digest seaweed. Is that even possible, or does this happen over many, many generations? And that became a question that I went after for years and years and years without a ton of success, until I met Mathieu Grousson and Mathilde Poyer. They came to my lab, and they really wanted to build these collections of microbes so we would get the individual isolates. And to that point, we had mostly just been looking at the DNA. They were able to put out this Broad microbiome library, which was very comprehensive for industrialized populations and pretty well characterized. You can still go in there. It's a great resource for longitudinal time series of 16S, for metagenomics, for metabolomics. There's tens of thousands of bacterial strains that are preserved. And we started to, we sequenced all of the genomes that came out of it. And then Tammy Lieberman, a postdoc, and she took under her wing Jay Zhao, a graduate student in my lab. They started to look at those genomes and say, can we see something about how these bacteria are adapting? And so one of the first things they looked at is, they would isolate the same bacteria, the same lineage over and over and over from the same person. And then they could see how deep was the phylogenetic tree, and then compare that to molecular clocks. And they estimated that there was about one SNP per year. And so it looked like most of the bacteria that they were looking at, most of the lineages inside people, had been in those people for anywhere from years to decades. And they were able to identify a lot of the genes that were under positive selection, because these genes would get the same mutation or maybe different mutations in the same gene over and over again. So maybe the same bacteria in 2 different people, or 2 different lineages separately got a mutation, or the same gene got multiple mutations. And when they looked at those mutations, they tended to be enriched in non-synonymous amino acid changing mutations, which is a signature of adaptive evolution. And they were able to use genomics to look at some of the dynamics of what was happening. So here, if you look at the x-axis as time, you can see that there's a blue variant, a blue strain variant that starts to take over. And then we could look at those genomes and say, well, what's going on in that blue strain variant? And here's an example of one interesting finding. So many of the findings they found were individual point mutations, SNPs. But here's an example where they actually found entirely new genes that arose. And we went back to the metagenomics and we found that, guess what? Even though that gene wasn't in the isolates on day 0, the gene was in the metagenome, which meant it was in another species. And ultimately, we were able to isolate that bug, and we found what species the gene had come from. And that was very exciting. So it was one case of this smoking gun. Well, Mathieu and Mathilde continued to build their collection. And they noted, as we've heard a couple of times already, that a lot of the critical biodiversity in the microbiome is disappearing rapidly, and it's found in indigenous populations. And they formed the Global Microbiome Conservancy and established that to preserve this microbial biodiversity. And they formed it on a number of different pillars. So the samples and the isolates, they are owned by the participants in the study. It's a nonprofit organization committed to capacity building and also sharing all the data and isolates with the rest of the scientific community. So they've been able to sample in a number of different locations, I think in at least 34 at last count populations. One of the interesting things is when you cluster them together, you can see there's kind of a gradient from the most rural to the most urban. Okay, so let me now tie everything back together. So here was my critical question: is are we getting these genes during the course of our lifetime? Are the bacteria in our guts picking up new genes? And this is the experiment. It's really simple. If bacteria in our guts are constantly picking up 2 genes, then if I pick 2 bacteria out of my gut and I say, what's the probability that they share a horizontal gene transfer, that they've exchanged DNA, it's going to be higher than if I pick 1 bacteria from my gut and 1 bacteria from your gut, right? Because the 2 bacteria from my gut, even though they're different species, they've been right next to each other, like, together in my body for presumably an amount of time that is less than my lifetime, right? So this was the question we were able to ask when we had gone through and isolated all of these different genomes from all these different populations as part of the Global Microbiome Conservancy. And the answer was really striking. So for every pair of different bacterial species, the rate of horizontal gene transfer was much higher within people than between people. And actually, we were able to break this down for different lifestyles. And we found that in rural, non-industrialized populations, actually the rate was much lower. And this hot spot of horizontal gene transfer in and on the human body is really a unique and new feature of urban industrialized populations. So I'll kind of end it there, and I'll say that one very new addition to the story as of last week is we got together. So Jay Zhao, the graduate student that Tammy had trained, worked with David Waits over at Harvard and Wenshan Zhang, and they developed a new microfluidic technique to get tens of thousands of bacterial genomes at once in a process that we call Microbe-seq. And we were able to use that to now get genomes, infer horizontal gene transfer from those genomes, and then compare that to the horizontal gene transfer we had inferred from the isolate genomes themselves, and it came out quite favorably. So we're pretty excited about that. And I'll thank folks again, and now it's time for the panel.
So while we're getting settled, I would ask that the house lights be brought up. And folks who do want to ask a question, you can assemble in front of these two mics. And what we're going to do is we're going to start with a question for all of our panelists. Now, Maria did do her homework, so she's going to have complete fiat to do anything she wants with her question time. So the question is the following. It's actually, there's an A part and a B part. You only have to choose to answer one, okay? So what excites you the most when you think about the human microbiome and its changes? Or what worries you the most? You only get to answer one of those. And we'll start with Justin.
I'll go with the worry. I think the questions that I posed at the end, I mean, I think it's very clear that we've changed our microbiome fundamentally with our lifestyle, and yet I think it's very unclear how this has impacted our health, just because there's, so many things happening with lifestyle change, and that could be affecting our health. We know that disease rates are on the rise. I mean, there's a lot of arrows pointing in the same direction, and so the concern for me is that, the diseases that are, killing us, populating our hospitals, are diseases that take decades to develop, and they're super hard to study, especially from the standpoint of prevention. And so if there are keys of modifying the microbiome, either the microbiome that we have by changing what we feed it, or by adding functions and species back to it that can fundamentally improve our health, I think that's going to be incredibly difficult to show, and it's going to take a long time. And so I think a lot about how can we accelerate that. And I don't have good answers, but that's one of my big concerns.
No, that's a good one. Gloria.
I think I'll go with the worry also, and the answer is very similar. I get very worried that we have impacted so much our biology. We haven't accepted that we are who we are because this is how we evolved. So we don't understand well enough our biology to respect it. And how are we going to maintain being high-tech societies, because that's what we want, and respect biology, especially in early life, which is when you have to respect biology because that's the crucial window of development, and then cheat nature when we are old because we don't want to die. So how do we manage those? How do we see life respecting the biology at every stage? And then especially at early life, how can we prevent? And I think we have to go back to natural ways and invent ways. How can we be natural and highly technological?
Yeah, no, thank you for that. And Eric.
I guess I'll have to go with excite, right? Because we've heard two worries. As a scientist, I'm so excited about the fact that we know so little about the microbiome. I don't think we know what the limiting nutrient is in the gut. Like, that's crazy. And the thing that excites me from the other aspect, from the Microbiota Vault and the Global Microbiome Conservancy and the initiatives that Justin is part of, is that if we want to, and we should, conserve the Amazonian rainforest, we have to conserve the Amazonian rainforest. If we want to preserve the microbial diversity associated with human beings, we just got to get out there and get those samples and isolate them and put them in the freezer, and then we're done. We don't need to preserve vast amounts of habitat. It is a tractable problem. It needs to be funded today because we may only have like 5 or 10 years to go out and get all those samples, but it's tractable. And to me, that's really exciting, right? I think everybody should be excited about that right now, because now is the time to get out and do it.
Yeah, great. Thank you for that, Eric. So good. Okay. So when you ask your question, why don't you first introduce yourself? And why don't you tell us where you're coming from? And if you want to tell us if you're a student or what you do for a living. And you might want to just take your mask off just for asking the question, and you can put it back on just because it'll sound better through the microphone. So we'll start over here.
Okay. Hello, my name is Katharina Richard.
Actually, can you point it up to your mouth? Yeah, get close. There you go.
My name is Katharina Richard. I'm from the University of Maryland. I work on vaccine development. This is my introduction to the microbiome. As I was listening to these talks, I was wondering about the effects of famine, displacement, war, major changes in populations on the microbiome.
Yeah, great. So the effect of major displacements of humans due to all sorts of terrible events. Who wants to start?
I could
Like
start
Gloria?
by saying that we know that stress greatly affects the microbiome, and in early life it leads to effects in the structure of the microbiome. We live in a world where we were naive to think that wars were over. It's still happening. So my prediction would be that there will be a lot of impact on the microbiome of those people, especially babies, children.
Yeah. Any other thoughts?
One other quick thing is just, I think it really matters how long and how severe the perturbation is. I mean, one of the things that has been stunning to me over the past probably 5 years or so, several studies showing that the microbiome can change really profoundly, but then rebound, whether it's antibiotic treatment, some of the dietary changes that we've seen. There's incredible resilience of the gut microbiome. And so severity of the perturbation and then, the multi-generational aspect of it, I think are probably important for how durable the changes are that are seen.
You want to add anything, Eric?
Not really. I mean, I would say there's probably big effects, but I mean, the fact that there are famines and wars and all these things, I mean, it's honestly, it's probably a secondary effect. I mean, there's other bigger problems.
Yeah. All right, we'll go here.
All right, I'm Nina Salama. I study Helicobacter pylori up at the Fred Hutch in Seattle. And I was really intrigued, Eric, by this last little observation that you shared, where the amount of gene transfer was actually less in the, I guess you could consider a more ancestral type of lifestyle than in the urbanized lifestyle. And that made me think about things that all three of you said, sort of suggesting that this big perturbation of urbanization that we've had is really causing a strong selective pressure, because as we know, horizontal gene transfer is a big driver of evolution. So, so it's kind of interesting to think that our microbiomes really are on tremendous pressure to change right now. So anyway, yeah, just maybe further comments on that. Individual organism level.
Yeah, I've thought a lot about that, of course, and some of it may be that there's been a huge increase in industrialized populations in genes that simply facilitate horizontal gene transfer. And a lot of times you see antibiotic cassettes riding around on that. So maybe in, the need for these microbes to pick up resistance to new antibiotics, they've also picked up a lot of machinery to move genes around. And it could be something as simple as the fact that in some of the smaller rural villages, many people have a lot of the same species. They will eat a lot of the same dietary fibers. Those different strains of the species will have a lot of the genes already that they need to eat a more, I won't say a more homogeneous diet, let's say day to day, but from person to person, more homogeneous for sure. Whereas in, big cosmopolitan city, you've got people coming from all over. And so there's just more genes to pick and choose from. So it could be something as simple as that. I think we just have no idea what it means yet.
Would either of you like to add a comment? No, that's great. So we also have folks participating remotely. So Jonathan, do we have a question from our colleagues who are in Zoom land?
Yes, we do. We have several. The first one for Professor Sonnenburg. Can you summarize the best approach to keeping my adult microbiome healthy and robust? And how is this any different than the post-antibiotic disturbance period? And in brackets, maintenance versus treatment strategy dependent on probation period.
Okay, Jonathan, we're having a little trouble hearing you apparently. So can you please say it, get a little closer to the mic?
Sure. Can you summarize the best approach to keeping my adult microbiome healthy and robust? And how is this any different than post-antibiotic disturbance period? And maintenance versus treatment, I think is what they're trying to get at with their question.
So how to keep your microbiome healthy and when you get perturbed by, let's say, antibiotics?
Yeah, it's a great question. I think there's pretty good consensus in the field that I said during my talk that a high fiber diet is probably one of the best things you can do for your microbiome. I think that why we saw negative results in our study or limited results is that a high fiber diet for a short period of time doesn't do a lot for you. But I think a habitual dietary, dietary fiber diet, you'll end up recruiting microbes that can degrade the dietary fiber. And certainly we saw the people with the highest diversity do very well on a high fiber diet. I think fermented foods have a long evolutionary history with humans dating back to when we were great apes actually, and fruit would fall to the forest floor and we'd eat it. There's evidence in alcohol dehydrogenase evolution that we've been consuming fermented foods for millions of years. So I think fermented foods, staying away from antibiotics, but if you have to take antibiotics, coming back to eating these foods that are rich in microbes and rich in dietary fibers to help build back the community. Those are my major things. I think there's a sanitation thing that I don't really, I believe, but I don't want to comment on in the throes of a pandemic. I don't know if either of you want to comment on.
Yeah, I mean, I think autologous restoration will come. If you have a baby that is healthy and you bank the poop of that baby, the day the doctor prescribes an antibiotic, it should be possible to restore after the antibiotic with the autologous sample. It's not been done, but I don't see why we are not moving in that direction.
Okay.
Hey, Justin, when you were talking about the fermented foods, one thing I was wondering is, do you think some of the benefits come from just the short chain fatty acids that are like in the food, or?
Generic, and you know, we've started to do this reverse translation in mouse models, trying to fractionate. This is the work of Sean Spencer in my lab, a GI fellow, and he's starting to take apart fermented foods, sterile filter them, just take some of the metabolites like lactate, and we are seeing that you can reconstitute a lot of the effects of a fermented food with just the filtrate, the sterile filtrate of the fermented food. So it really suggests that the metabolites may be doing a lot of the work that we're seeing.
Just to clarify, that diversity in the fermented food does not engraft in humans, right?
Exactly, and I just hit on that briefly, but the increased diversity that we see, only about 5% of that is are microbes from the fermented foods. Don't know where the microbes are coming from, whether they're blooming from under the level of detection in the microbiome or coming in from the outside and engrafting, but they're regular members of the microbiota that show up. Yeah. So
I'd just like to editorialize that the idea of just having a capsule of extracts from fermented foods versus actually eating the fermented foods, that's depressing to me. I'd much rather eat the
totally
exciting fermented foods that we were discussing earlier.
I'm the total opposite, Steve. I'd rather have the capsule than the fermented foods.
Well, you know, you have the busy life of a physician, Dr. Kraft.
Colleen Kraft, Emory University, lifelong learner. This kind of work really excites me, and I think about it a lot. I think some of the things that I'm looking forward to, the discovery that I'm sure you all and hopefully many students in this room will obtain, is what are some of the surrogates for healthiness. So we know that life expectancy in some of these resource limited settings is much lower than ours, right? So that doesn't improve necessarily with gut microbiome. So do we all want to be in a resource non-limited setting with a HADSA diet to try to have long life expectancy? Do the parasites and some of the other things that occur in some of these resource limited settings? I've actually eaten caterpillars as a delicacy. You know, do some of these things help their guts become what they are? And then So essentially, resource-rich setting with a HADSA microbiome, is that what we're aiming towards? What are the surrogates of sort of that health? Obviously, we talked about BMI and all that kind of stuff. So hopefully that was clear.
I mean, I think what your point, we definitely don't want to go back in time. I think we want the best of both worlds, and figuring out what those factors are, I think remains to be determined. But I think that you're right in that there are certainly a lot of aspects of the industrialized lifestyle that are beneficial, and antibiotics are a perfect example of this, but a lot of the other medical treatments and advances, nutrient-dense food. I will say, you know, one of the really interesting things here is that our brains are wired for caloric density. And so, you know, if you ask the Hadza what they prefer eating, it's not the fiber-rich foods, it's meat and honey. And yet to get honey, they have to they have to create fire, they have to climb a tree with smoke, risk their life, smoke bees out of a tree hive, throw honeycomb down to the ground. You know, if we made our kids do that every time they wanted a cupcake, they would eat many fewer cupcakes. And so I think we have to think about how to balance how we're wired with the ecosystem that we live in. I'll take a question from here.
Hello, my name is Tanay Cardona, come from Imperial College London. I'm very interested in molecular evolution and genome evolution. Quick question to Eric. You study horizontal gene transfer, but from the perspective of genome evolution, I wonder if you also suspect an increase in gene loss, perhaps by deletion, or maybe an increase in the emergence of pseudogenes. Is that something that you have looked into? Do you speculate?
Can you repeat the last part?
Do you, in the same way that you see horizontal gene transfer, would you expect gene loss by deletion or the emergence of pseudogenes? Is that a possibility? Have you looked into it?
Changes in gene dose or number of genes in an organism.
So, it's hard to say because the variation in genome size is so large. I don't think we've been, so maybe if I can just kind of paraphrase the question, if the amount of horizontal gene transfer is going up since industrialization, are we going to see an increase in the size of those industrialized strains compared to, let's say, their ancestor strains? The amount of variation there is from strain to strain is so large. I don't think we can test that hypothesis yet, but there's certainly a large amount of gene gain and gene loss, and most of these recently horizontally transferred genes, they don't stick around very long. They get lost from the genome and then they come back. They tend to live in those genomic islands that are really variable. So I think they're going to change as, you know, the selective pressures are there to maintain them or not.
Okay, thanks. Jonathan, do we have a question from an online participant?
We do. This one is for Dr. Dominguez. Thank you for focusing your work on mothers and babies. What can we do to increase diversity and biodiversity for microbiome research in this population?
Jonathan, we're still having a little bit of trouble.
Oh, really?
Yeah, hearing you on the stage. I apologize.
It's okay. This one is for Dr. Dominguez. Thank you for focusing your work on mothers and babies. What can we do to increase diversity and biodiversity for microbiome research in this population?
To increase the diversity of the research.
Oh, what can we do to increase?
What can we do
the research
to increase
on diversity
and research?
On diversity research.
I think the question was referring to diverse populations. How do we get more diverse populations into your research, into this research?
How to bring more diverse populations
human
into
population.
the research.
Yeah,
there we go.
That's a very good point, because it's very hard, as Eric was saying, it's very hard to get funding when you're going to do studies that are global. And we need to convince our agencies that we need to change that. The world is one little house. Things are happening simultaneously in many places, and we are all going in the same boat. So definitely we should have more representation of microbiome studies from other parts of the world. I mean, it's highly dominated by US, China, but in order to understand really human biology, we need to be inclusive of other human populations.
Great, thank you. All right, we'll take a question from here.
Hi, thank you. Yes, this is for Dr. Dominguez as well. I'm Amy Edwards. I'm a pediatric infectious disease doctor at Rainbow up in Cleveland, Ohio. I think I might be the only pediatrician in the group. Love your work, fantastic. I was wondering, I saw a study recently that showed that the bacterial population in breast milk for women who pumped was significantly different than the breast milk isolated directly from the breast of the woman. What advice, so as a pediatrician and as a PEDS ID doc, what advice would you tell us as clinicians to give to our moms who have to pump because, you know, America sucks, like to help with infant diversity, you know, when they have to pump in order to not lose their job?
I get these very good questions. I get a lot of questions similar to that by email. And my advice is, first, don't feel guilty when you have to impact because you had to have a C-section or because you need an antibiotic. If it wasn't for that, either you or your baby may not survive. So we see that in the jungle, you know, it's heartbreaking to live without medicine. So don't feel guilty and appreciate medicine. And then there are many natural ways that are restorative. And breast milk is an amazing restorative factor. I tell the mothers, just kiss your baby, skin to skin, lick your baby, you know, breastfeed, etc. Just be natural with your baby. And we don't understand all the ways of restoration yet.
Question here.
Hey, I'm Austin Campbell. I'm a PhD student from the University of Michigan, Tom Schmidt's lab. I was just interested, this question's for Dr. Sonnenburg, but also Dr. Dominguez. Your view of the disappearance of microbial taxa over time and changes in the microbiota over time often takes a view that's sort of hundreds, or in the case of the Paleofacies, even thousands of years long. But one of the figures that Dr. Sonnenburg showed in terms of the increase in potentially microbiota-associated diseases and inflammatory issues, the timeline started at 1950. So I was wondering just if you guys could talk about any attempts to sort of bring those two timelines into sync and maybe get still a correlation, but a closer correlation between the changes in the microbiota and what we have medical records for in terms of these diseases.
I have problems knowing when one word ends and the other starts, sorry. But is the question about the loss of the microbiota from the 1950s?
Yeah, I'm sorry. It was just about basically synchronizing the timelines of the changes in the microbiota, which is often you guys looked at things that were actually either thousands of years or hundreds of years ago, or sort of implied that time scale of development. But the disease information we have is more recent. Dr. Sonnenburg showed a figure since 1950. So I was wondering about just bringing those two timelines together and kind of lining up that data.
I mean, it's the thing that's
Paleo, you know, with the
exactly. I mean, it would be great to have a stool bank that had sampled people for the past 200 years, and you could go back and do like this, you know, longitudinal or even cross-sectional across that time. But I think maybe the key for doing that is if somebody knows of a stool bank like that, because there may be old hospitals that have old stool samples from, you know, that would be amazing. But you know, I think tracking populations as they transculturate, I think, is probably the best way to go forward with that sort of question, and then do the, you know, epidemiology at the same time that you're doing microbiome sampling. But I think it's a great point. It's just like a big disconnect between when we have disease data and the timelines we're talking about for microbiota change.
Most of the diseases really rocketed from the mid of the 20th century. And that is antibiotics. That's when antibiotics came to be used. And our studies in the Amazon show that just 1 year of exposure of very remote villages that were not exposed before is enough to see decrease in diversity. Antibiotics are potent antimicrobials.
Okay, we're gonna have time for 2 more questions. So I'm gonna take one from this column and then one from this column. So.
Hello, can you, Sam DeGregory, based at UCLA. I was really fascinated by all 3 of your talks. Particularly, it seems like there's a lot of data showing how stable the gut microbiome is. Like, it's hard to repopulate lost species. But then on the flip side, you know, there's this horizontal gene transfer happening. So my question is the gut microbiome stable or is it plastic?
Right. So Justin's answer is yes.
Yeah, my answer is yes to that. I don't know. You know, that I think you know, one of the keys or one of the ways that I think about it is it really, I mean, it was nice, the array of data today, because it really captures kind of how I think about the microbiome, which is there's, you know, this adaptability to it. And we certainly saw that in the Hadza over seasons, with microbes disappearing and then coming back when different foods would come back the following year. So, you know, it may, and the evolvability of being able to incorporate gene cassettes that are involved in accessing new nutrients. I mean, what a cool way to extend your biology really rapidly, right? So this is in some ways an incredible technological platform for adjusting to changes in our environment. But I think that as I mentioned, that malleability can be taken to an extreme with perturbations that, you know, our biology and our microbial biology has never seen, you know, historically through the millennia. So I think that there probably is some amount of plasticity that is intrinsic and built into the system, and or at least that we've co-opted to our advantage, and then that can go off the rails really quickly. So that's the way I think about it. And I think the mechanisms of resilience are probably also built into the system, whether it's bacteria living in crypts or in the appendix that repopulate after perturbations. I think there are elements of the system that are there for backup to provide stability over perturbations, adaptability, but then things can go awry if you have big perturbations.
Yeah. I would add, there is still host selection. So your microbiome will not be subverted by your dog's microbiome. Their host selection is very strong, and I think that puts limits to the plasticity and keeps the microbiome host specific.
I think that there is a large turnover of strains and species in the microbiome, but, you know, one study I didn't get to talk about, but was really my first study in the microbiome, was just tracking my own microbiome over the course of a year. And I got salmonella poisoning, I got food poisoning during that study, and it wiped out an entire phylum. It wiped out almost all of my firmicutes. Before I got salmonella, my firmicutes were at about 38% of the total biomass. And after I recovered from salmonella, firmicutes were at about 38%, but they were replaced with entirely new species. So you know, and it was down to like 38 or 39%. It was, you know, it was really, really close to what it was before. But my physiology, my gut had rebuilt an ecosystem that was very, very similar with new pieces, right, with new species. So I think there's aspects that are robust, but the individual strains and species they may have a higher turnover.
Thanks for that. And the last question.
Well, thanks so much. My name is Lucas, and I'm from the University of Tennessee, Knoxville. And I wonder if I can ask 2 questions.
What's that?
Can I ask 2 questions?
No, you can only ask one question.
Okay.
Yeah, one per customer.
Yeah. So you show beautifully that there is a huge difference between biodiversity in industrial societies compared with hunter-gatherers. So I was wondering, just looking at hunter-gatherers, if you compare Inuit populations with Amazon forests, for instance, if there is a difference in latitude, like if in the tropics, you would have to find more biodiversity in microbiome compared with like in high latitude.
So is there a relationship between the biodiversity in a particular area?
Yeah. And looking at hunter-gatherers,
versus what we see in the microbial diversity of the individuals there.
Ecological gradients in diversity, do they also
Yeah.
apply to human microbiome populations?
Is there
Yes.
correlation?
I don't know. I don't think anybody's looked at that.
And I think it's a difficult question to ask because there's not a lot of sampling of indigenous populations that are out of the tropics. So I think I've seen hypothesis papers on this before, and I think it's a good point and one that needs to be looked at, but I'm not sure of data that speaks to it.
By how industrialized versus how rural they are. So I don't think, even if there was a correlation, I'd be skeptical that it was really the latitudinal gradient in diversity that you see in other ecosystems. It's just too confounded with the industrialization.
Your analysis nicely shows that regardless of geography, it's lifestyle, right? In the urban cloud, it doesn't matter if it was Asia or Europe or the savannah jungle people. So I think it's a lifestyle is what really drives, it's a major factor driving.
And
which is a bit of a surprise.
But you know, so it may still correspond because definitely like the diversity of foods that are available in the tropics are probably much broader, much more diverse dietary options than like, you know, the Inuit or something like that.
Yeah.
So there may be lifestyle factors that are dictating a gradient like this. Which
I think your question raises a really interesting point that the assumption, our default might be the more ecological diversity there is, there's going to be more diversity, but that these human associated factors of lifestyle are going to really dominate and drive it more. And that really seems to be where the data is taking us. Now, thank you for your question. So I'm going to end with a comment, not a question, because we do have to stop. But Eric's personal experience raised one of my other questions. Originally, the question was, you know, is there an optimal human microbiome, or is this even a question we should be asking? And something I've been spending a lot of time thinking about is, does it matter who the organisms are in a particular microbiome versus what activities or services are being provided by those organisms? And your example is, you know, it's not the species that might be as important as the metabolisms associated with those species. And I think as we move forward, that's the hard question, right? Because it may not matter who is making enzyme X, as long as enzyme X is getting made. And that all of these relationships with our own human microbiome and our own human physiology are going to depend on the production of those kinds of, you know, services. Does that sound okay? All right. It's a hopeful note, though, for future funding. Okay. I want to thank you all for participating, but let's give a giant round of applause for our panel. I hope you all have enjoyed this session as much as I have. And I wish you all a really great rest of conference. Enjoy the rest of ASM Microbe and have a wonderful evening. Thanks, everybody.